Hello, my dear students. Welcome you all to Shri Jayendra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the last lecture, we discussed about the mechanism of conduction of impulse through a nerve. I will repeat the last lectures, the last and the final slides so that you can recapitulate the content again, as this part of the chapter is very important, which involves nerve conduction. So what exactly nerve conduction is, we got to know in the last lecture. So I am just recapitulating the contents, the spatial points, so that you can frame the you know uh, the topic. The first thing is stimulus. It is a change which can generate any action potential. Okay, so it is a change which can generate an action potential, right? Okay, stimulus, if we talk about threshold stimuli, so it is a stimuli of minimum strength which is required for excitability of nerve. Okay, stimuli of minimum strength that can generate an action potential. See, when we talk about the resting membrane potential, RMP, resting membrane potential, okay, and when we talk about action potential, so what exactly that mean? Resting membrane potential means the nerve, when we talk about the extracellular compartment and intracellular, mean in the extracellular fluid of a nerve, the sodium ions, they are more in comparison to the intracellular, and the intracellular part will be having more amount of potassium ions in comparison to extracellular. The outer part of this membrane, neuroedema, is positively charged because of the accumulation of positively charged particles, cations like sodium, and the intracellular part is negatively charged because of negatively charged proteins. Proteins. Okay. So these negative charge proteins make the inner part negative. Inner part negative. Okay. So presence of negatively charged proteins make inside of nerve negative. Okay. Now, the moment this nerve it receives the message is received in the form of either chemical stimuli or a nerve stimuli. Okay, the nerve stimuli can be through the sensory organs or the sensory receptors, whether it is proprioception, whether it is olfaction, whether it is vision, anything. So the membrane at resting stage means suppose the resting membrane potential is minus 70 millivolts. I'm talking about a skeletal nerve. Okay. So the resting membrane potential is minus 70 millivolts. The moment receives the stimuli, there are leak channels, there are sodium gated channels on the membrane. These channels will open. Okay. As in the next previous slide I have shown. Yeah. The resting membrane potential was suppose minus 70 millivolts. If I am talking about the skeletal nerve, means skeletal nerve means a nerve which is related to neuromuscular junction. Okay. So this nerve is having a resting membrane potential of minus 70 millivolts. The sodium ions are accumulated on the outside, and these are the leak channels. You can see these all channels are protein gated. So these channels require an enzyme for the transportation of sodium and potassium. So in the presence of these, especially the ATPase enzyme, because this is a process which requires energy. Okay, so this is an active process. Okay, so active process. So the potassium ions goes outside. When the potassium goes outside, so the number of potassium ions on outside increases. Okay, and because of this, the polarity of inside region changes. As the polarity of inside region changes, okay, what will happen? Now the nerve becomes a point of attraction for the impulse because change in amount of charge will lead to change in or creation of potential difference. And because of this potential difference, the charge or the so-called current or microcurrent will flow. Understood? Okay, got it. So this is the resting membrane potential when we talk about, okay, potassium ions go outside. Okay, as the potassium ions go outside, the inner part of the membrane changes. Okay, and the sodium ions move inside. Okay, so this efflux and outflux helps to change the polarity of the membrane. Okay, I'll show you. First, I'll write for you all or none law. This is a very important law which is favored by muscular contraction as well and the nervous contraction as well. Although nerves they don't go contraction but they go excitability. Okay. All under law says the moment the threshold stimuli is received, okay, on receiving a nerve fiber will conduct an impulse. A nerve fiber will conduct an impulse. So suppose for the conduction of impulse, there is a minimum requirement of a threshold potential of 45 millivolts. Potential difference is always in negative. Okay, 45 millivolts. So this 45 millivolts needs to be, it should be there the moment any stimuli is conducted. All the stimuli which are having the potential difference lesser than 45 millivolts, they won't be able to conduct their message, they won't be able to conduct themselves. So either the nerve will conduct all the messages above the threshold stimuli or it won't conduct. So it is all or none law. It is not that the message will partially be conducted. Okay. The stimuli will completely be conducted if it is having a potential above that threshold stimuli. And it also says that the nerve, the moment it receives a stimuli, okay, it is not going to take the other stimuli the moment it is carrying, the ongoing stimuli. Means a stimuli is passing through the nerve, it is passing through the nerve, okay, it is on the way, and at the same moment, if you want, That the stimuli it moves from this side to this side. Now the stimuli is here, and another stimuli. No, no, it cannot be done. It cannot. It is just against the rule of nervous conduction. Okay. So now, so the extracellular fluid is having more amount of Na plus. Okay. There is a technique which is known as what, cathode ray oscilloscope. Okay, which is cathode ray oscilloscope. Positively charged particles are outside, means sodium. Okay, negative charge particles, proteins. Now, a question comes that the negative charge particles like chloride ions are also there. Okay, when we do, it's not like that we are having only negatively charged proteins. No, we do have chloride ions, we do have negative ions or the anions, but the fact is proteins, because of their structure, they can have more amount of free charge, right? Protein, because of their structure, they're three-dimensional, their quaternary structure, they can store, they can, on their surface, they can show multiple free charges, free ions. Okay, so that is why it is considered to be negative, because of presence of these proteins. Okay, the nerve cell is having a resting membrane potential of minus 70 millivolts. I have already told you. Okay, the potassium ions are more outside. The potassium ions are more inside, and sodium ions are more outside. Okay. Now, the process of leak channels is like this. Suppose these are the guarding proteins, transporters. Okay. These are the phospholipid molecules. Okay. Now this is suppose Na plus K plus ATPase complex. Okay, and there are some channels which are known as what, as I have already told you, gated channels. Okay, they have a gate like structure or a cap like structure. Okay, so these are known as what, gated channels. And these are leak channels. Leak channels, they don't have any cap all the time. So these leak channels, because of the change in polarity of the nerve, these leak channels, they are basically guarded by proteins. So proteins, they can change their conformation, they can change their three-dimensional structure in the space, and because of which the potassium ions goes outside. Okay, potassium ions goes outside. This is the way that how the potassium ions and sodium ions they move. Now, the leak channel, sodium and potassium moves along with the con along with the concentration gradient. Concentration gradient. That is, along with concentration gradient. In leak channels, there is passive diffusion. We can say the sodium and potassium ions, they move along concentration gradient. But when the reversal is done, I mean, once the depolarization is done, then it's okay, the action potential is achieved. But at the moment, at the cost of repolarization, when it is to be reversed, mean potassium ions needs to come inside from outside. Then it requires a action potential. Then it requires then it requires a active energy. Okay, or the energy from ATP enzyme. Okay, then it is a active process. Okay. The resting membrane potential is more permeable to K plus. Okay, that is why K plus is leaked out more. Okay, so more leakage, right? So it's about for three potassium ions, two sodium ions come in, go outside. For two sodium ions go inside. Okay, this process is done with the help of sodium pump, that is a combination of a protein that is okay. It's three sodium ions and two potassium ions. To maintain the electrical neutrality, as K plus means the membrane is more permeable to K plus, so K plus will leak out more. Okay, so when K plus will leak out more, so this sodium potassium pump will basically help to reverse the condition. Okay, so as the K plus is moved out more, their charged the potential difference is more. Okay, so that is why we require more sodium ions to go outside. Okay, to balance the electrical neutrality. It is basically ATP is converted into ADP and inorganic phosphate. Okay, the threshold stimuli, it is around 55 to 60 millivolts. Okay, the process of depolarization is basically opening of sodium voltage gated channels. Depolarization involves opening of sodium voltage gated channels. Okay, which helps in the influx of movement of rush of Na plus. खूप सारा Na plus means it comes down. Okay, like this. The moment Na plus comes inside, what will happen? The negative part becomes positive. Okay, because of influx of sodium ions. Okay, and the outside part becomes negative. Okay, other part maintains its electrical nature as previous. Okay, so it is step by step process. It is not that the whole nerve is, receiving the stimuli. It is getting under the process of depolarization at the same time. Okay, so inside of the membrane it becomes positive and outside becomes negative. This is known as what? Action potential. Okay, now when we talk about the repolarization, during depolarization the voltage gated potassium channels open. So, which leads to efflux of K plus. Efflux of K plus. Okay, like this. So it's positive, therefore, okay, negative. Okay. Inside becomes negative and outside becomes positive. This is repolarization. Now, hyperpolarization. As the membrane is having more permeability for K plus ions, so due to more efflux of K plus ions, inside becomes intensely negative. Okay, so this is hyperpolarization. Understood? This is a very important term, refractory period. And what is the usage? It is a time period between two consecutive nervous stimuli or nerve conductions. Okay, it is a time period under which a second stimuli cannot generate nerve impulse. Cannot generate. Okay, this is a refractory period. Okay, so we understood about depolarization, repolarization, resting membrane potential, everything. Okay. When we talk about the minimum refractive period, the minimum refractive period is basically 1 by 1000 part of the second for an axon. Okay, so refractive period is by 1 by 1000 of a second for axon. Nerve conduction is always unidirectional. Okay, unidirectional conduction. So here you can see that we have nodes of Renvir over here. So conduction through nodes of Renvir is known as what? Saltatory conduction. This conduction is fast. Okay, these are nodes of Renvir. The myelin sheath is made up of basically fatty layer. It is a fatty tissue layer, which is non-conductor or insulator. That is why the impulse, when reaching to the axon hillock, it jumps from one node of Renvir to the other node of Renvir because node of Renvir is basically not having the myelin sheath covering. So this conduction is known as what? Saltatory conduction. Saltatory conduction, which is faster, around 120 meters per second. Okay, it is seen in vertebrates. It is seen in vertebrates. So this is the neuron anatomy in which you can see the dendrites, okay, having multiple branches. The rough endoplasmic reticulum, ribosomes are there, Okay, Golgi apparatus is there, nucleus is there, nucleolus is there, membrane, nuclear membrane is there, microtubules are there, mitochondria is there, smooth endoplasmic reticulum is there. All are present except centrioles. Okay, then microtubules, neurofibrils, neurotransmitter receptors, everything is. This is synapse. Okay, synaptic vesicles. These are the synaptic vesicles. These balloon-shaped synaptic vesicles. This is synaptic cleft. The space is known as what synaptic cleft. Okay, and these are mode of Ranvier. Okay, as we have studied in muscles, the same thing we have already studied in nervous tissue, the epineurium, perineurium, endoneurium, like that. Okay, now repolarization. With the increase of sodium ions inside the nerve cell, the membrane becomes less permeable to sodium ions, where the permeability membrane, the membrane of the permeability of membrane to potassium ions increases. Okay. So during the process of depolarization, the permeability of sodium ions or the sodium leak channels open. So with the increase of sodium ions inside the nerve cell, the membrane becomes less permeable to sodium. Okay. Okay. Now you have to step sodium, and it becomes more permeable to potassium ions. The sodium ions are pumped out of the cell, and the potassium ions are pumped into the cell until the original resting state of ionic concentration is achieved. Okay, so once the nerve comes in in the phase of action potential, it moves back to the phase of resting or tries to back to the resting membrane potential. So this makes the membrane negative on inside and positive on outside. This process is called repolarization. Okay. The last movement of ions is thought to take place by an active transport mechanism called sodium potassium pump, also called sodium potassium exchange pump or sodium pump. The sodium potassium pump is process of expelling out sodium ions and drawing in potassium ions. Because of the efflux of potassium ions more to the outside, it needs to bring back, and that is why we need a protein gated, channel or a way to make the potassium ions to re-enter the space where they previously were belonging to. Okay, so because of this the process becomes active. Previously it was going through the passive diffusion because the movements were undergoing through the along the concentration gradient, but here the potassium ions, they are forced to move back to the original space, and that is why they require the sodium potassium pump. Okay, the entire process of repolarization required some time during which the nerve cannot be stimulated again. Okay, the entire process of repolarization, it required some time during which the nerve cannot be stimulated again. This period is called refractory period. Okay, the moment nerve is under the process of stimuli, it is under the process of action potential. It cannot make or it cannot take another impulse to make that impulse to reach to action potential. At a particular time, there is only one impulse which can reach or which can cross the threshold stimuli and reach to action potential. During repolarization, as the cell returns to its resting potential, the neuron is ready to receive another stimuli. Okay. Now there is a very important fact. If the refractory period is shortened or it is overlapped, suppose there are two stimuli A and B. Okay. And there is a third stimuli C. C is basically threshold stimuli. Please try to understand this example. So there is no question about C that it will go for conduction. Yeah, it is appropriate when it is appropriate for conduction, and it will go for conduction because it already have crossed the limit, which is the minimum limit which is required for the conduction. Now A and B, if they are very close to C, if they are very close to C, or A and B, they are competitive inhibitors of C. Okay. A and B are competitive inhibitors of C, or we can say A and B are some poisons or some accelerators which can diffuse or help in removing out excessive number of ions from the intracellular fluid to extracellular fluid or vice versa. What I'm trying to say is, if A and B, the potential rise very close to C. So there is high chances of A and B to pass along C. The moment C is conveyed by a nerve, the nerve says, stop, stop, stop. I will conduct one at a time. I will carry one at a time. So nerve will ask A and B, do you have the threshold potential within you? So A and B says, yes, we are the part of threshold potential. So now there is a confusional state of nerve that whether the nerve should allow C or A and B. So these are the conditions which are, you know, creating a sense of dilemma, delusion, confusion. Mean the moment we are in a state of not taking a proper decision, when we are in a state of confusion. It may be due to change in hormones, deficiency of hormone, change in nerval conduction, or deficiency of neurotransmitters, whatever is the reason. But the scenario, the sky which is required, the platform which is required for these conditions is this only: overlapping of more than one stimuli, overlapping of threshold stimuli. Okay, so now coming to the next slide, the synapse. Okay, now before moving to the synapse, I would like to add some more points. Okay, that the speed of conduction is more in myelinated nerves, as you know. Okay, the diameter of axon, when we talk about the diameter of axon, the larger the diameter of axon, more is the speed of conduction. So, what are the factors? Factors affecting the speed of conduction, nerve conduction. What is presence of myelin sheath? Presence of myelin sheath. Second, large diameter. More will be the diameter, more will be the conduction. Okay. When we talk about like giant squid, it is the largest, it is having the largest diameter neuron. Okay. The temperature of conduction is more in warm-blooded animals. Okay. So temperature also can affect the velocity of conduction. Conduction is more. Okay. Now, synapse. It is an area of functional contact between one neuron and another for the purpose of transferring information. Very important, functional contact. It is not the physiological or anatomical contact. It is a functional contact. Synapses are usually found between the fine terminal branches of axon of one neuron and the dendrites or the cell body of another. One of the neuron ends with the synapse, the fine terminals, okay, and the other neuron starts with its dendrite over that area. Okay, this type of neuron is called exo and dendritic synapse. Okay, Sir Charles Sherrington in 1861-1954 was the first person who used the term synapse to the junctional points between two neurons. Okay, so it is a junction between two neurons. Charles Sherrington, he gave the term. Okay, now when we talk about the types of synapses, okay, exodendritic, as we have studied in the previous slide, exodendritic. So it is the most common case, most cases. Then exosomatic. Somatic means cytoplasm. Okay, and axon with axon, exoaxonic. So these are the different types of synapses. So basically, synapse is a chemical junction. It is a functional junction. It is a chemical junction. Okay. So you can see the ending of the nerve. It basically ends up in a knob or synaptic knob, which is having synaptic vesicles. So these are synaptic vesicles. Okay, and this is a synaptic knob, which is a bulb-like structure. Okay. Here we are having a presynaptic knob. This is the synaptic cleft. This is the synaptic cleft. This is the presynaptic knob. And this is the dendrite of postsynaptic neuron. This is the axon of presynaptic knob. Okay. And this is the dendrite of postsynaptic neuron. Okay. Now, movement, these are synaptic vesicles which are filled with neurotransmitter. Okay. This is telodendrium or telodendrite of presynaptic neuron. Okay. Now, these synaptic vesicles are filled with neurotransmitters. Vesicles are filled with. Okay. So the chemical synapses are more advanced than electrical synapses because it is both. It helps in both excitation and inhibition. Okay. So these synapses are chemical synapses. Okay. It is a type of chemical synapse. Okay. So the synaptic vesicles, they are filled with neurotransmitters. The movement, the nerve impulse, it comes through this way. Okay. So it leads to efflux of calcium ions. These calcium ions, these accumulation of calcium ions will basically make the solution hypertonic. Okay. So I will let you understand. Afterward, after this discussing the structure, I will make you understand. Structure of synapse. A typical generalized synapse consists of a bulbous expansion of a nerve terminal called presynaptic knob lying close to the membrane of a dendrite. The cytoplasm of the synaptic knob contains mitochondria, smooth endoplasmic reticulum, microfilaments, and numerous synaptic vesicles. Okay, so it contains mitochondria, synaptic knob, it contains mitochondria, endoplasmic reticulum, microfilaments, and numerous synaptic vesicles. Each vesicle contains neurotransmitter, a chemical substance responsible for the transmission of nerve impulse. The membrane of the synaptic knob nearest to the synapse is thickened and forms the presynaptic membrane. The membrane of the dendrite is also thickened and called postsynaptic membrane. These membranes are separated by a gap known as synaptic cleft, which is around 200 angstrom, okay, across. The postsynaptic membrane contains large protein molecules which act as receptor sites. The postsynaptic membrane they contains large protein molecules which basically act as a receptor sites for neurotransmitter and numerous channels and ports. Okay. These membranes are separated by a gap known as what synaptic cleft, which is around 200 angstrom across. The postsynaptic membrane contains large protein molecules which act as a receptor sites for neurotransmitter and numerous channels and ports. Okay. The two main neurotransmitters in vertebrate nervous system are acetylcholine and noradrenaline. Okay. Although other neurotransmitters also exist, very true, acetylcholine was first neurotransmitter to be isolated and obtained by Otto Loewi in 1920. So acetylcholine was the first neurotransmitter to be isolated and obtained by Otto Loewi in 1920 from the endings of parasympathetic neurons of the vagus nerve in frog heart. Okay, so Otto Loewi in 1920, okay, he obtained this neurotransmitter acetylcholine from the endings of parasympathetic neurons of vagus nerve in frog heart. Neurons releasing acetylcholine are described as cholinergic neurons, and those releasing noradrenaline are described as adrenergic neurons. Okay, as the parasympathetic nervous system is basically concerned with only and only acetylcholine, while sympathetic nervous system it basically is concerned with both acetylcholine and noradrenaline. Okay, mechanism of transmission of nerve impulse at a synapse. That's what I was talking about. The process of chemical transmission across synapses was discovered by Henry Dale in 1936. Okay. The physiological importance of synapse for the transmission of nerve impulses was established by McLennan in 1963. A brief description of the mechanism of synaptic transmission is given below. When an impulse arrives at a presynaptic knob, mean calcium ions from the synaptic cleft enter the cytoplasm of the presynaptic knob. Calcium ions, very important, from the synaptic cleft enter the cytoplasm of presynaptic knob. So when the impulse reaches or arrives at the presynaptic knob, the calcium ions they move from the synaptic cleft and they enter the cytoplasm of presynaptic knob. The synaptic cleft is around 10 to 20 nanometers. Okay. Okay. Yeah. The calcium ions causes the movement of synaptic vesicles to the surface of the knob. Now the calcium ions they move, okay, and because of they move and they also help to move the synaptic vesicles to the surface of the knob. Okay, the synaptic vesicles are fused with the presynaptic membrane. The synaptic vesicles are fused with the presynaptic membrane and get ruptured. Okay, these synaptic vesicles are fused with the presynaptic membrane and get ruptured. That is exocytosis to discharge their contents, that is neurotransmitter, into the synaptic cleft. Now, what exactly is happening? The calcium ions they cause the movement of synaptic vesicles to the surface of the knob. The synaptic vesicles are fused with the presynaptic membrane and get ruptured. That is exocytosis. To discharge their contents, neurotransmitter into the synaptic cleft. Okay? Is it okay? The synaptic vesicles then return to the cytoplasm of the synaptic knob where they are refilled with the neurotransmitter. Now the synaptic vesicles they return to the cytoplasm after exocytosis of the synaptic knob where they are refilled with neurotransmitter. Okay, so this is the chemical impulse which is or the chemical synapse which is more advanced. Okay, which require neurotransmitters. Now, yeah, an action potential arrives and initiates synaptic transmission. Okay, sodium channels open depolarization of the axon terminal membrane. You can see the opening of sodium channels. Depolarization of the terminal membrane causes voltage gated calcium channels to open. The calcium channels voltage gated they open. The calcium ions enter the cell and trigger fusion of the acetylcholine vesicles with the presynaptic membrane. You can see the vesicles they are fusing with the membrane. This is the fusion of the vesicles to membrane. You can see. Okay, and they releases acetylcholine. Acetylcholine molecules diffuse across the synaptic cleft and bind to the receptors on the postsynaptic membrane. They move from here and bind to the postsynaptic membrane. Okay. Activated receptors open chemically gated sodium channels and depolarize the postsynaptic membrane. The spreading or depolarization fires an action potential in the postsynaptic membrane. Acetylcholine in the synaptic cleft is broken down by the enzyme acetylcholinesterase, and the components are taken up by presynaptic membrane for resynthesis. After synaptic transmission, acetylcholine vesicles are recycled. The same process is recycled again. Okay, I hope you got it. First, what will happen? The sodium channels will open, which will further open the calcium channels. The calcium channels come here. They will make the solution hypertonic. The vesicles will fused. The vesicles will be carried by these sodium channels. They will get fused to the membrane. The content will come outside. This content basically helps in the depolarization of the postsynaptic membrane, which further helps to open the sodium gated channel. The sodium channels open and the nerve conduction starts. Now, there is an important term that in chemical synapse a neurotransmitter is required, okay, and the synaptic cleft is 10 to 20 nanometers. But when we talk about the electrical synapse, there is no requirement of neurotransmitter and the synaptic cleft is 2 nanometers. Okay. The neurotransmitter of the synaptic cleft binds with the protein receptor molecules of the postsynaptic membrane. This binding action changes in the membrane potential of the postsynaptic membrane, opening channels in the membrane and allowing sodium ions to enter the cell. Already understood. This causes depolarization and the generation of action potential in the postsynaptic membrane. Thus, the impulse is transferred to the next neuron. Having produced a change in the permeability of the postsynaptic membrane, the neurotransmitter is immediately lost from the synaptic cleft. In the case of cholinergic synapses, acetylcholine is hydrolyzed by enzyme acetylcholinesterase, which is present in high concentration at the synapse. The products of hydrolysis are acetate and choline, which are reabsorbed into the synaptic knob, where they are resynthesized into acetylcholine using energy from ATP. Okay. So the process of acetate plus choline into acetylcholine is an active process. It is an active process. Neurotransmitters, as explained in the discussion of the synapses, neurotransmitters are the chemicals released from a presynaptic neuron that interact with the specific receptor sites of postsynaptic neuron. Yeah, they are released by the presynaptic neurons and they go to the postsynaptic neuron. The synaptic delay, which is very important to understand the inhibitory and excitatory neurotransmitters, the synaptic delay is around, it is lesser than 1 millisecond. There are about 60 chemicals thought to have have capacity as a neurotransmitter. Okay, so the major excitatory neurotransmitters in brain and okay, when we talk about the major excitatory neurotransmitter of brain is glutamate. The major excitatory neurotransmitter of brain is glutamate. Okay, epinephrine, adrenaline. Epinephrine, adrenaline, they are the most, you know, we can say more patent than not epinephrine and noradrenaline. So epinephrine and adrenaline, they are more patent, they are more in use, okay, in comparison to norepinephrine and noradrenaline. So neurotransmitters, they are basically acetylcholine, made up of acetylcholine, or made up of glycine and GABA aminobutyric acid, or aspartate and glutamate. Okay, aspartate and glutamate. So acetylcholine, they are basically made up of biogenic amines. Okay, I'll write in the next page. Create a page. Wait. Yeah. They are biogenic amines. They help in brain and memory pathways. They act as a neurotransmitter in brain and memory pathways. Mainly a part of parasympathetic neurons. having a calming effect on body. Relax. Okay. It also helps to activate neuromuscular junctions. The neurotransmitters having glycine as amino acid, okay, or glycine and GABA. They are the types of neurotransmitters only which act as a neurotransmitter. GABA is a gamma aminobutyric acid, which is an inhibitory neurotransmitter. Inhibitory on brain. Okay, glycine is secreted spinal cord. Okay, now the inhibitory neurotransmitters includes gamma aminobutyric acid and glycine. Gamma aminobutyric acid in brain and glycine in spinal cord. Okay, understood? Acetylcholinesterase helps to cleave it. Acetylcholine cleaved by acetylcholinesterase. Okay. Then comes the aspartate and glutamate. They are also excitatory. Brain. They are also amino acids. Alzheimer's disease is because of decrease in acetylcholine. Decrease in acetylcholine. Okay. Huntington's chorea disease because of decrease in GABA aminobutyric acid. Gamma aminobutyric acid. Okay. Then there is a one more neurotransmitter, dopamine, which is a catecholamine. Okay, and noradrenaline and norepinephrine. Okay, so dopamine, which is a catecholamine, it is a neurotransmitter in midbrain and limbic system. Okay, means the emotional brain. The motor neurons, because of dopamine, gets the inhibitory effect, and the limbic system it gets the excitatory effect. Motor neurons, because of dopamine, gets an inhibitory effect, and limbic system gets an excitatory effect. Okay, this is about the dopamine, which also act as a now excessive of dopamine and decrease of dopamine. They are also very important, cofactors or imposing factors of the different diseases like Parkinson's and schizophrenia. Okay, I will explain. I will explain in the coming slide. No issues. Synapse is a one-way valve. The synapse cannot transmit an impulse in the reverse direction as the dendrites cannot secrete a neurotransmitter. Thus, the synapse acts as a one-way valve, allowing the conduct of impulse from axon to dendron. Yeah, that's true. No need to elaborate this point. Okay, it's already self-explanatory. Now, synaptic delay. The transmission of impulse across a synapse is slower than its conduction along a neuron. Very important. Okay. This is because of the time needed for the release of neurotransmitter, its diffusion through the synaptic cleft, and its action on the postsynaptic membrane. The difference in the rate is called synaptic delay, which is around half a millisecond at a body temperature of 37 degrees Celsius. Synaptic fatigue. A repeated stimulation of the presynaptic node may deplete the neurotransmitter, and this may fail to stimulate the postsynaptic membrane. This condition of the synapse is termed as synaptic fatigue. It lasts for several seconds, during which the neurotransmitter is resynthesized. Synaptic fatigue is the only fatigue that affects the nervous tissue. Conduction of the nerve impulse along the neurons is not subject to fatigue. Okay, that's very true. All or none law I've already explained, so I will not be explaining. You can go through the text. Okay. Now, yeah. Now I will explain. Because of excess of dopamine, a condition named schizophrenia. Schizophrenic. Okay. Characterized by hallucinations leading to psychotic behavior. Characterized by hallucinations leading to psychotic behavior. Okay. Then decrease in your loss of dopamine, Parkinsonism or Parkinson's disease. Okay. Characterized by uncontrolled shaking. Okay. Norepinephrine or noradrenaline is also catecholamine, which is released by sympathetic nervous system. It increases the metabolic rate, heart rate, respiratory rate. Okay. Now everything has already been explained to you. Okay. Yeah. So that is all for my dear students. I have completed this chapter. This last slide and these slides I have just given for your view, for your revision. Okay. So please go through these slides. These are the tips and tricks. Okay. You can go through. Okay, the chapter is finished from my side. We are left with the question and answers, and we will do, as I've already explained, we will do all the question and answers at a once at a stretch. Okay, for all the chapters of physiology. So till then, bye bye. Take care. Stay safe. Stay healthy, everyone.